School in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video, so we are continuing regarding phases of cell cycle. Okay, phases of cell cycle, our knowledge point of view. So there are 3 phases of cell cycle we can observe. So what are the main important points we can be discussed means, so here the phases of cell cycle point of view. So what are the phases of cell cycle we have seen? So there interphase is there, M phase is there, again in interphase no. So there G1, yes, G2, so we are observing, isn't it? So there in phases of cell cycle point of view, so 2 phases are there. So 2 basic phases there. One is interphase and second one is M phase, isn't it? So again in interphase 3 stages, G1, yes, G2, isn't it? So now G1, yes, already we have seen, but somewhat information to be discussed related to the S1, S phase. So there our knowledge point of view, what is information we can be discussed in S phase means, once you see, one more time you see, phases of cell cycle 2 types, one is interphase and one more is M phase. So in interphase no, again our knowledge point of view we have been divided into 3 stages. So there G1, yes, G2, very technically we have learned in previous video, isn't it? So now in previous video itself we have discussed regarding G1, yes. So somewhat information to be discussed in this video regarding G2, regarding S1 and G2 complete information we have to be discussed in this video. So now after completion of G1, yes, G2, definitely we will move forward to learn the information of S phase, isn't it? So thereby in interphase, so G1 we completed, yes information, let us continue to discuss. Now in animal cells, during the S phase, important, even a NEET examination point of view also important. In animal cells during S phase, DNA replication begins in the nucleus. Correct only no? So in animal cells, during S phase, DNA replication begins in the nucleus. So and the centrioles duplication takes place in the cytoplasm. My point you understood. So in animal cells and plant cells, no, up to here correct only. DNA duplication or a DNA replication process begins in the nucleus. Up to there, both in animal cells and plant cells correct da. But here centrioles duplication, that is only we can be seen in animal cells. Centrioles duplication takes place in the cytoplasm while S phase is going on in nucleus of animal cells. Point you understood. So that's why to avoid the confusion, what is the information we have written here? In animal cells only we are concentrating. In animal cells no, during S phase. So S phase means synthesis phase, synthesis of DNA no. So that's why that particular point we can be taken as DNA replication. So begins in the nucleus. Definitely DNA present in the nucleus. That's why duplication of DNA taking place in the nucleus. While that process is going on, in cytoplasm centrioles duplication process also takes place. That is the intention of author to explain this information. Next one, during the G phase or G2 phase. Now, so that is our information regarding G2 phase of interphase. So proteins are synthesized in the preparation for mitosis while cell growth continues. So their cell growth process is going to be continued. While this kind of a process is going to be taken place, what is happening, Amma? So in G2 phase, protein synthesis or proteins are synthesized in preparation for mitosis process. So thereby, what are the proteins which are required for the process of mitosis? All these are all kind of proteins are prepared in the G2 phase. So in old information, so what is the main important point we will discuss means, listen carefully. So what are the activities? So one more time you see there, in G2 phase, we are discussing, we are learning one point, proteins are synthesized. So in preparation for the mitosis, likewise we are discussing, so means what are the proteins which are required for the process of mitosis? All kind of proteins, all kind of proteins will be synthesized in the G2 phase. So thereby here what is the main important point means, so what are the main activities or what are the metabolic activities are started in the G1, all kind of same processes will be continued even in G2 phase. Okay, so thereby even in a G1 phase also one point we can conclude, in a G1 and a G2 phase definitely proteins are synthesized. Rest. Next one, some cells in the adult and this important. So last year and before last year, they have asked one neat question here. Okay, listen carefully now. Some cells in the adult animals do not appear to exhibit division. So means, so what is the point here we are understanding? Suppose if you take some animal cells do not appear to exhibit the division. So in some cells, there is no chance to the cell division process. So for example, heart cells means here continue to study, so thereby you will understand, and many other cells divide occasionally as needed to replace the cells that have been lost because of the injury or cell death. What is the meaning? In animal cells, some cells all the time will not be divided. Will not be divided all the time. Only certain means only divided once you see at the spotlight. Once you see, divide only occasionally. Occasionally means if the needed, if the cells replacement is there means then only the cells will undergo for division. Point you understood means like a normal cells madri, what are some kind of cells means, for example heart cells. So what are the cells are present in heart cells? All the time these cells will not be divided. Just these cells will be divided occasionally. Suppose requirement irukka appo mattum divide agum. Suppose requirement illiya, at the time will not be divided. Point you understood, ma? So thereby here, so when these cells are needed, definitely replace at the time, that particular cells are divide and replace the cells that have been lost. Lost means lose, we know very well. So means, so here then only when the cells by getting of injury or suppose if the cell death point of view, so these kind of activities, so because of these processes, if the cells are lost in that particular area means to fill that particular gap only cells undergo for division, that is also occasionally. Point puriyadama, so nalla save pannikonga. Suppose in animal cells, for example heart cells if you take there, suppose if needed, suppose cells new cells are needed means then only, so some kind of part cells will be divided. Suppose there is no need means those kind of cells will not go for division. So means here, for example, some examples point of view, cell division takes place occasionally. Point purida. So that is a point here we are understanding. These cells do not divide. Once you see one more time, these cells do not divide further. So again once the suppose if the particular need is fulfilled, suppose now there is no any requirement means at the time what is happening now, these cells do not divide further, get exit from the G1 phase. So once G1 phase ki enter avana kandipa S phase, G2 phase, apra M phase kandipa taken place. That's why what is happening, once the need is fulfilled, at the time these cells will not be entered into the will not be entered into the interphase. In interphase only, no, G1 yes, G2 we will observe. Will not be entered into the G1 phase, S phase and G2 phase. Definitely get exit from the G1 phase. Am a point puridam. Suppose if you are here means diagrammatically or very by using of either one particular method I can explain, but in absence of you explaining, no, so it is difficult only to understand. But my dear students, if you have interest to listen this, to learn this, this one you can understand, amma. So one or twice definitely go through this, so thereby kandipa you will understand. So one more time I will remember, ipa paathukama. Suppose if the cells do not divide further and get exit from the, from not there, but we can read this, get exit from the G1 to enter into the inactive stage known as quiescent stage or G0. So in a previous video, one diagram, cell cycle diagram we have drawn, G1 yes, G2 aprom prophase, metaphase, anaphase, telophase, cytokinesis. So together M phase, aprom one cell edthi, aprom finally two cells, one round diagram we drawn, there G0 phase also we have mentioned. We drawn, no? So there I mentioned G0 phase also. So thereby here whatever the cells are there, these cells without entering into the G1 phase, so coming back to the or moving into the inactive stage is known as quiescent stage or G0 stage. I hope you understood this. Right. Now the cells in this stage remains metabolically active. Sir, if these cells, why sir these cells are not dividing? What happens sir means these cells are metabolically active da. Metabolically active only, but like suppose if you take one bike, no, bike neutral on panmana on, but it will not be running position, isn't it? Suppose gear potavana, suppose if you operate a first gear means then only it will start to move. So likewise here also, what are the cells who are present in a G0 phase, no, metabolically active da. But when the cells are required, when new cells are required, no, then only what are the cells are metabolically active, no, those cells start to divide. Example puriyadana. So likewise even here in these cells also, so metabolically active da, but when the cells are required newly, no, then only these cells will be divided furtherly. I hope you understood this. Next one, but no longer proliferate unless called on to do so depending on the requirement of the organism. So we have discussed the explanation on this. Next one, in animals, so here also one need bit irukra. So in animals, so cell division is only seen in the diploid somatic cells. Point puriyadara. One more time once you see. So in animal cells, cell division only seen in the diploid somatic cells. Diploid somatic cells means, so definitely 2n. Diploid means 2n condition. Somatic cells means body cells except means here the exception n reproductive cells. Reproductive cells means cells which are involved in reproduction. So those cells there. So thereby here one point let us follow. In animals, this kind of cell division only seen in the somatic cells. That too, in animals point of view, diploid cells only. Diploid cells, 2n we will take now, n, 2n, 3n likewise. So diploid means 2n stage. Remember panniko. However, there are few exceptions. One point we are learning, in animals, no, somatic cells, that too 2n diploid cells only cell division takes place, or this kind of mitotic cell division takes place we are discussing. Again now author going to give one exceptional point. However, there are few exceptions to this where the haploid cells are divided by mitosis. For example, male honeybees. This is important. This is one exceptional point. Next one, against to this. So here in plants point of view, what is information we can discuss means, in plants can show mitotic divisions in both haploid and diploid cells or diploid organisms. Means in plants point of view, so both mitotic division and both mitotic divisions we can observe. So in plant cells, mitotic division we can see in both type of cells. Now you can take this point. So mitotic division can be seen in both in haploid cells as well as in diploid cells. But in animals point of view, So this kind of cell division we can observe only in diploid somatic cells mostly, but here one exceptional point. But in plants point of view, mitotic division that occurs both haploid cells as well as in diploid cells. Okay, so next one, let us start to discuss the M phase. M. So in this M phase, so what is the information here we can be discussed means in M phase point of view. So M phase there, so the most dramatic, 1 minute, so there most now you see, so M phase most dramatic period of the cell cycle. It is most dramatic period. Why? Because our knowledge point of view. So this particular phase, actually here I didn't mention. Why? Because I would like to give that particular point through my voice. Okay, so simple M phase for our convenience to study, let us divide it into two types. One is karyokinesis and one more is cytokinesis. Don't worry, already this explanation I have given in a previous video. Now M phase for our convenience to study, we are dividing into two types. One is karyokinesis and one more is cytokinesis. If karyokinesis means division of nucleus. Again in karyokinesis, no, prophase, metaphase, anaphase, telophase, likewise of 4 stages are there, no. Upon cytokinesis means next stage after completion of karyokinesis, cytokinesis only, no. There in cytokinesis, what are the points we are discussing? The division of cytoplasm we are discussing, isn't it? No worry, I am not going fast, just I am giving explanation only, so no worry, you can understand. Okay, so now let us start to discuss. So M phase is the most dramatic period of the cell cycle, involving so a major reorganization of the virtually all components of the cell. So here it is a most dramatic period of the cell cycle. So there major reorganization, major reorganization virtually what it is, the components of the cell, so going to be reorganized in this stage or in this period. So thereby, since the number of chromosomes in the parent and progeny cells in the same. So here, even though means here this major reorganization of the components is going to be taken place in the cell now, definitely here one point we have to remember, that thing is number of chromosomes in the progeny cells in the cell will be same. So that's why this particular stage or this particular mitotic stage we will be taking as by one more name is known as equational division. M phase is also known as equational division or mitosis. Point purida. So though for convenience, so now here they are dividing. So for our convenience to study the mitotic process, now we divided into 4 stages. 4 stages of nuclear division. Means here 4 stages of nuclear division. Means nuclear division means karyokinesis. So this particular karyokinesis, for our convenience to study, we divided into 4 stages. So next one, before start to discuss the prophase, metaphase, anaphase, telophase, so somewhat information we have written here, that is also once let us read and learn. So it is very essential. So which one? Karyokinesis. Karyokinesis is very essential to understand that the cell division is a progressive process and very clear cut lines cannot be drawn between the various stages. So there it is a kind of progressive process and very clear cut lines. So very clear cut information will be there and diagrammatically cannot be drawn between the various stages. Okay, next one, karyokinesis involves, now learn panikonga. Karyokinesis now for our convenience to study we can be divided into 4 stages. So first one is prophase, second one is metaphase, third one is anaphase and fourth one is telophase. That's why dots I have mentioned, second one, third one, fourth one likewise. So now let us start to discuss the prophase 1. Okay, now this particular prophase 1, which is a first stage of karyokinesis, this ellarkume theriyum. So first stage only, no, that one no need to write, but I have written, so right. Now prophase 1 is a first stage of karyokinesis of mitosis, follows the S and G2. இப்போ in interphase now, first of all G1 is there, next one G2 S is there, next one G2 is there. இப்போ G1 S G2 அப்புறம் in M phase no karyokinesis only no, in karyokinesis prophase only no, first one. That's why this particular prophase follows the which one and which one? S and G2 phase. Amma the S and G2 phase means thereby prophase stage that follows the S and G2 phase of interphase. Now here one important point, in the S and G2 phases the new DNA molecules are formed or not distinct but intertwined. So means here in this particular case now we have discussed. So what is the point we have been discussed here? G1 and G2 phases, if you observe there more definitely, so the new DNA molecules of formation we have observed, but that is not distinct. Okay, that one we cannot be seen very clearly, but it is intertwined, they are saying. Okay, next one, prophase is marked by the initiation of the condensation of chromosomal materials. This is important point. Now here in a prophase point of view, sir, if these many lines are there, so what are the main important points we have to remember under prophase? Means here we have written, once you see these two underline lines are there, no, prophase stage is marked by the initiation of condensation of chromosomal material. அப்படினா so முன்னாடி chromosomal material எப்படி இருக்குன்னா, that is seems to be like thread like chromatin material will be there. That particular thread like chromatin material start or initiation of condensation of chromosomal material. Initiation of condensation. So by the process of condensation and all, finally in the end of the prophase, no, so finally converted into chromosomes. Next one, the chromosomal material becomes untangled during the process of chromosomal condensation. So means here while this condensation process or the chromosomal condensation process is going to be taken place, so this particular chromatin material will be untangled. So definitely untangled. So finally next one, in the next point, what is happening? The centrosome, this particular centrosome helps in the development of bipolar spindle fibers. So spindle fibers means in previous chapter, no, we have seen no microtubules. Remember, microtubules means doublets, triplets, likewise we see, no. So likewise here also some kind of tube like structures, microtubules. Apadana micro means chinnadi, tubules means chinna tube like structures formation takes place with the help of centrosome, so which helps in the pulling of chromosomes. So ipo botany point of view, bipolar spindle fibers, so did you hear the term? So no problem, I will tell you in the next stage only. Now we are studying a prophase, no. In the next stage we will discuss about the bipolar spindle fibers. So those bipolar spindle fibers are formed by the centrosome. It is a kind of cell organelle which helps in a cell division. Okay, now let us see that point. Centrosome which had undergo duplication during S phase of interphase. So centrosome, so definitely centrosome, centrioles are there, no. In animal cells we have seen no centrosome. Adema one diagram like rangoli madri, one diagram is there, no. Triplets we have seen. Ange ulle when you observe the centrosome structure, na ulle rendu centrioles are there, one pair of centrioles which are arranged perpendicular to the each other like 90 degrees angle, I said, no. So that information so try to remember. Ipo centrosome which had undergone duplication during S phase of interphase, this particular point we learned already, now begins to move towards the opposite poles of the cell. So definitely once the duplication is completed now, so this centrosome definitely moves towards the opposite poles of the cell. So point purida appuram ipo sir one more, so actually main important points enna, so here one point I have written remaining points of prophase in the next video I will give you. Why? Because here no gap so try to understand, right? Now chromosomal material ipo paathukom, chromosomal material condenses to form compact mitotic chromosomes. Before we see no in prophase 1 what is happening, initiation of condensation of chromosomal material we see no, so that particular chromosomal material condensation process appadana condensation ana ipo very huge length of chromatin material irukana adi konju condense avum. Condensana suppose ipo cotton cloth irika new cotton cloth, so if you put in water for some time no tanninalle potuvana koncham shrink avum ema, so we will use the shrink no ade our botany point of view condensation word shrink avum, but shrink avum like a dress madare shrink avuna, so it will not be suitable. It will not be correct word to use, that's why so we are using this term. So thereby chromosomal material condenses to form compact mitotic chromosomes. Ipo chromosome that like chromatin material no adi chinna konchu short avum short ayi final by empty or by end of the prophase no, so that will be converted into chromosomes. Okay ma, now last and final point in this video, chromosomes are seen to be composed of two chromatids attached together at the centromere. Ipo rendu don't forget one neat bit idi, suppose if you take one particular chromosome no, there kandipa two chromatids are present. Okay, well, so all the time definitely we will observe the two chromatids in one chromosome, exception. So one exceptional point ipo telophase we see no. So in a telophase we have seen no ma ipo based on the position of centromere chromosomes are divided into 4 stages no. In the previous chapter we learned metacentric, submetacentric, acrocentric, telocentric. Ipo in a telocentric only one chromatid is present, but remaining all types of chromosomes no, definitely two chromatids are present. Idu adi nalla paattukoma சரியா, நல்லா ரிமெம்பர் பண்ணிக்கோங்க. ஸோ மீன்ஸ் டெஃபினெட்லி இஃப் யூ டேக் அ ஜென்ரல் ஸ்ட்ரக்சர் ஆஃப் குரோமோசோம்னா தேர் கண்டிப்பாக டூ குரோமேட்டிட்ஸ் ஆர் ப்ரெசென்ட் அண்ட் தீஸ் டூ குரோமேட்டிட்ஸ் ஆர் அட்டாச் டுகெதர் அட் தி சென்ட்ரோமியர். ஸோ ஐ திங்க் ஸோ திஸ் லைன் இஸ் நாட் அப்பியரிங் டு யூ, ஸோ த்ரூ மை வாய்ஸ் யூ கேன் கெட். Okay, two chromatids are attached together at the centromere. Okay ma'am, so up to here let us save in our mind. So remaining points let us learn in next class. Okay ma'am, so in this class what is information we have discussed, so somewhat remaining information regarding S phase we discussed and G2 phase we discussed, apron G0 phase we discussed. After G0 phase information now, so where this particular cell division takes place, so that we have seen. Apron M phase we have discussed, in M phase also, so prophase 1 we have discussed. So this is a small information regarding today's video. So continuation information let us learn in next class. Thank you ma, listen carefully and be in good position in our subject. Thank you.